We're already in Chapter 9. So Chapter 9 is called Molecular Biology. So biology, molecules. Well, actually, it's one molecule that we're talking about. One molecule that seems to be more important than all of the other biological molecules in its size and in its impact. And so really, what this chapter is about DNA. You can think of it that way. And how we copy it, what happens to the DNA. But the field is called molecular biology. It's kind of interesting. The biology of DNA. Yes, question?
Is it also called biotechnology?
No. Biotechnology is another chapter.
Okay,
That's about DNA also, but that's more like engineering. This is kind of the natural, what happens naturally. And they're definitely related. So DNA, it's universal. Every living thing has its code in DNA. Things like you've never seen before, like I just wanted to find some strange-looking invertebrate creature. This can be found in sewage. It's a gastro trick. I don't know. I think they're kind of cute. It's an invertebrate animal. Plants like this ivy, chinchilla, I used to own one, our pets, ourselves. Everything is coded by DNA. Every living thing on the planet. Every living thing that we know of. Maybe this will change when we discover life on different planets or meteorites or something. But as far as we know, every living thing is coded by DNA. And the sequence of DNA was worked out relatively recently, 1953. Not the sequence, the structure of DNA is what I mean to say. So that's relatively recently that we've even understood what DNA is composed of. And you brought up biotechnology already. How much has come since 1953? We not only understand it now, we're beginning to control it and design it the way we'd like. But let's give you a little bit of history on how and why people figured out. These scientists figured out the way it was. Rosalind Franklin has to be given a lot of credit for this. She did work in X-ray diffraction. And so what you do is you put X-rays upon molecules and you see how the X-ray wavelengths are diffracted when they hit the molecule. And so just showing you some of the data that she got, This to me would mean nothing because I'm not an X-ray diffractionologist or whatever you call it. But for her, it's like, she figured out that the genetic information, which they were able to isolate, the genetic information must have the shape of something like a spring or a corkscrew because it would be that shape, and sending X-rays upon that would give a pattern like this. And so that was a huge help for researchers that followed. There's something like a spiral that's going on here. And then this handsome gentleman, Erwin Chargaff, he was able to, again, they knew genetic material. They were able to isolate genetic material. But as far as the structure of DNA, he really helped in bringing us along on that. And it was, these scientists, they're so dedicated and patient because he went through and he isolated DNA from the nucleus of ox, humans, hens, salmon. You can read the rest of the list. A lot of different organisms, even bacteria. And what he did was they knew that the hereditary molecule was composed of adenine, guanine, thymine, and cytosine, and they could actually separate those out. But because he did this detailed work, and he was like, well, how much adenine? How much cytosine? How much guanine? How much thymine? And then he was able to get a pattern. So just look at the data. Let me give you a minute just to look at the data. And so try and interpret what's happening here. See a pattern? Is there a number that comes up? Yes. The number 1. Okay, so where do you see the number 1 popping up?
All over the place. All of them are pretty close to 1, like in the last.
All of them?
In the last column.
In these last 2 columns here. On this one as well. These numbers are not 1, well, that one's 1 exactly, but these numbers are right around the number 1. So that's the ratio. So the ratio of adenine was about equal to the ratio of thymine, and the ratio of guanine is about equal to the ratio of cytosine. But if you compare thymine to cytosine, you don't see that 1. Does everyone see the pattern now that it's pointed out? Almost all right about 1 right here. So how he interpreted that was that the amount of adenine in DNA is about equal to the amount of thymine. The amount of guanine is about equal to the amount of cytosine. Another huge clue. And the reason I bring up Rosalind Franklin and Erwin Chargaff is that when you learn about DNA and DNA structure, you always hear about who? Yes. You hear about Francis and Crick. But, they couldn't have... I'm sorry, I said Francis and Crick. Watson and Crick. James Watson and Francis Crick. They couldn't have done the work that they did without those 2 clues. The helix shape and that adenine is about equal to thymine, cytosine is about equal to guanine. So what they did was they created a model. And you can go to the Science Museum in London, a free museum. We had to get to London. You can actually see, here I am checking it out, you can see their model. People have likened it to like Tinker Toys. Anyway, they created this model with the information that they had. Okay, we know it's going to be a helix. We know that adenine and thymine are about the same and cytosine, guanine are about the same. So they came up with a model and information they knew about hydrogen bonding and chemistry and affinities between different molecules. And they were able to come up with this now very famous double helix. Interesting, because this is just a different day. If this was today, how would they be modeling? In the laboratory with little pieces of things? I'm guessing on the computer, right? We do computer modeling. Anyway, this is a different day. And they were able to come up with, they came up with the proposal that DNA consists of 2 long strands that are twisted into a double helix. And the 2 strands are held together by hydrogen bonds, which you guys know about. Remember, we talked about hydrogen bonds. They're not so terribly strong bonds that are attractions between the partial positiveness of hydrogens and the partial negativeness of other molecules, such as oxygen and nitrogen. So 2 strands, a very long molecule that's basically 2 strands, but it's twisted. And they realized that this was a great design for a molecule that has to do what it needs to do, basically to copy itself and also to provide information on how to build organisms. Because you want to be able to access it. And the fact that the hydrogen bonds are not so tight, that means that this helix, and we're gonna pull this apart in a couple of different ways. We'll see that it will open up occasionally. So that information can be accessed, but then it can close back up. So it was some brilliance on their part. And so the building blocks of DNA are nucleotides. And the nucleotide has a general formula to it. That is, it has a phosphate, it has a sugar, and then it has a nitrogenous base. So the phosphate, you can note here that there's a negative charge on it. The sugar's name is deoxyribose, and that's where that leads to the name DNA, deoxyribonucleic acid. So it's that sugar. And then the base, well, this is where the information part comes in, because that base can be one of four things: adenine, thymine, cytosine, or guanine. And they all have different structures, which are shown here. You don't need to memorize the structures, but just know that there's four different ones. And that's what gives the code of the DNA code. And so how they come together is the nucleotide. I'm going to circle a nucleotide here. Phosphate, sugar, that's the sugar, nitrogenous base. The nucleotides are end to end to end to end on one strand. And they will, through hydrogen bonding, attach to nucleotides on another strand. What's funny about the way this strand is written? It's upside down. And I purposely wanted to pick a figure that you could see that happening, because that's how it works. It's like, this is a nucleotide here, phosphate, sugar, base. This is a nucleotide here, phosphate, sugar, base. So it's anti-parallel. How they line up is anti-parallel. That'll be important for us later. But just, can you see that there? So we've got, this shows 3 pairs of nucleotides. And this shows also that the thymine is hydrogen bonding to an adenine. Thank you, Shargoff, right? And the cytosine is hydrogen bonding to a guanine. So they're anti-parallel. So then we need a way to talk about when it's going this way versus when it's going this way. And so the jargon that scientists use is 5', 3'. So what the heck does that mean? That refers to the sugar, and the sugar deoxyribose has 5 carbons to it, and so they number the carbons. They start here, 1, 2, 3, 4, 5. So this is the 3' end. Can you see which end this is? That's the 5' end. Up there, which I can't reach, it's the 3' end. And then over here is the 5'. So they're anti-parallel, like that. Any questions so far? Yes?
Is it like the guanine and cytosine, they have to complement each other? If there's a C, the other side would be guanine. If it's thymine, it would be adenine.
Yes.
So is there any chance of like be an error where it's like G and there's something else?
Yes. And can the body fix it? In most cases, yes. So we're going to get to that. We're going to get to mutation, and we're going to get to proofreading, which is going to be, you've predicted that. But we'll have to get there in a minute. Okay, so it's 2 strands, and it became apparent to Watson and Crick that this is really the perfect molecule for copying itself, as we know it has to do. We know the DNA copies itself in the S phase of interphase. So how does it do that? Well, it can do that because each side has the full complement of information. And so we can really basically pull these 2 strands apart, and each of the strands will have the information to be able to make its partner. So we're pulling it apart here, and we see that we've got a T exposed. Well, an A can fit in that spot. Over this side, there's an A, and what's going to pull up right here and bond with this A? T, thymine. And so it's like if I covered one side of this, it says TGAC. What's under my hand? A, C, T, G. You're brilliant. So having a molecule that has information on both sides, each of them can serve as what's called a template. Each one can serve as what's necessary to construct the other strand. And so when you pull it apart, like we see here, we'll be doing in DNA replication, when you pull it apart, then then we'll have the information to create another strand. So these two strands are identical. Do you see? Confirm that with yourself. These are identical. But they were once just one double helix that got pulled apart. So DNA is absolutely huge. It's really hard to fathom how big DNA is. It's been said that for just one of your cells, if you took the DNA from one of your cells and you stretch that, and your cell is like going to be maybe 60 micrometers. Your cells are tiny. And you took the DNA from that cell and you stretched it out, it would be 2 meters, which is taller than me. That's just from one of your cells. If you took it from all of your cells, your DNA could go to the moon and back several times. Do you see what I mean? It's really hard to fathom. It's absolutely huge. Because it's our storage, and so we need to have high capacity. So that also means that the cells are going to have to be very organized in how they keep that storage. They can't just randomly pick on my daughter for a second. Her way of taking care of a room is she just throws everything down on the floor. That's not a way that's going to help in retrieving things later, because it's very difficult, of course, if you don't have things organized. But it's extremely organized. And so having that huge amount, then it has to be very compact. And we have seen it get super compact as chromosomes, but even when it's not undergoing cell division, it's still compacted. It's just not as compacted. And so we see that what happens is that DNA helix gets wound tightly around histones, which are proteins that act like a spool. And so really a chromosome is not just DNA. It's also protein too. It's the proteins that the DNA gets wound around. And so then they're called nucleosomes at that point, and then that then gets coiled. And then that gets further condensed. And then when it's time for cell division, as we saw, that gets condensed even further. So they're in bundles. So it is a helix, it's true, but it's not like we've ever seen anything like this under the electron microscope, because this is way too small. What we've seen is bundles of it. So thankfully, there's a nice video about this that we can take a look at how it bundles up.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So pretty impressive what we're doing with our DNA to keep it all tidy. So any questions about that, about DNA and how it's organized, its appearance?
The bonds between, sorry, what do you call it, the nitrogenous base,
Mm-hmm.
is that going to be the same in mitosis and meiosis?
The hydrogen bonds between the bases is
One of them can only connect to a specific one?
is inherent in DNA. So mitosis, meiosis, it's irregardless of that. That's all DNA has A and T and C and G. And so I'm glad you're bringing up mitosis, meiosis because we're just coming out of that chapter. So in both cases, in the S phase of interphase, there would have to be an opening up of that and filling in of bases on either side to duplicate it. So yeah, that happens irregardless. Any other questions or thoughts? So DNA, molecular biology, there's two very important functions of DNA. One it has to do with making more cells on your body, or making gametes to produce offspring that you'll leave progeny here on the planet. So DNA making more DNA. So that's why I've got the arrow pointing back. So we call that DNA replication. If that didn't happen, then basically life would stop, and you wouldn't have any cells to replace all the ones that you damaged over your lifetime, which would be pretty ugly. And you wouldn't replenish the cells of your body and your organs. So we need to do DNA replication. But then there's another major role that DNA plays. DNA is the code of life. DNA is why I've got brown eyes and brown hair and no dimple. DNA codes for proteins, which makes up most of our body. Okay, mainly I'm a water bag, but after you take away the water, I'm mostly protein. And so DNA codes for that protein. So we need to do that too. We need to not only see how DNA makes more DNA, we need to see how DNA makes protein. And it's both in this chapter. We'll do the first one first, though. And it ties in well with where we have just been, DNA replication. So in DNA replication, entire genomes are copied. Each one of your cells has the entire genome, the entire genetic makeup. Each one of your body cells has the entire genetic makeup. So every time you make a new body cell, each body cell has to get the whole darn thing. And so that it's basically, it gets replicated. That's the word for it, replication. So it's self-replicating. And DNA is the perfect molecule to do that, like I said, since it has information on both strands. So in eukaryotes, this happens in the S phase, as you know. And so this, I just wanted to kind of tie this together for you. When we leave mitosis, after we do anaphase, chromosomes pull apart, telophase, they go to either end, nuclear envelope forms, cytokinesis, cell splits into two. Then at that point, the chromosomes are in this unduplicated state. We still have a pair. This represents a homologous pair, but they're not duplicated. So between ending mitosis and beginning mitosis again, we need to copy it. We know that happens in the S. And so we can't really see it like this, but this is the end result. We can't really see it because the DNA molecule is absolutely huge. And when it copies, it can't be bundled up because you have to access it. It has to pull apart the strands. And so it can't do that when it's all like tightly round. But anyway, when we end mitosis, it looks like this. Then it's kind of invisible, can't really see it. But then when they leaves interphase, they look like this. That's the duplicated state. Each of these are called sister chromatids, and they're identical to each other. So this is what we're talking about, just to tie it in with where we've been. So now I'm at the level of, let's take a look at that helix. So in order to make copies of the helix, we're going to need enzymes. We're going to need lots and lots of raw materials. All these here are meant to represent nucleotides. Can you remind me what 3 parts you find in a nucleotide? Phosphate, sugar, base. Phosphate, sugar, base. So that's what's represented. So we need tons of it. Where do we get it? We get it in our diet. We can't order it in from Amazon. We need to get it in our diet. So keep eating. And so that's where we get these these nucleotides. We need enzymes for this to happen. The enzyme that is doing this here, which is basically having it separate, is called helicase. Because the problem is that it's all twisted. And so you need an enzyme that's going to help it to relax so you can get at it. So the name is helicase. To be quite honest, there's other factors involved and other enzymes involved. But you're going to be spared that. I'm just going to kind of hit the highlights and try not to give you any untruths. It is more complicated than is seen here. But do know that helicase is an enzyme, and that helps it to unwind. Relax, DNA. Unwind a little bit. And then where this relaxation happens is called the replication fork. So, and the replication fork is the separation here of the 2 strands. Now, does the helicase start at any old spot? No. It starts at a specific spot that's called the origin of replication. And this is when I get to tell you that's what a replicon is. If you were wondering, what's with replicon? Replicon is another word for the origin of replication. So replicon is where DNA starts to unwind. And so as it unzips, it creates this replication fork. And so we're going to be acting on either one of these strands. But the first thing is to get it unwound a little bit so you can get in there. So I really like this diagram. I like the level of detail it gives us. And it reflects that we're talking about DNA from a chromosome. But of course, it's much more bundled when it's in the chromosome form. And this is color-coded. We've got adenine as red and thymine as orange, I guess. And they're always paired together. So you can scan and see there's no errors here. Cytosine and guanine go together. And that has to do with the fact that they form hydrogen bonds. Adenine and thymine, hydrogen bond with each other. Cytosine and guanine, hydrogen bond with each other. And so, so in order to get copied, what needs to happen is that the DNA needs to relax. So do you see the enzyme helicase and its role right there? So helicase is having the double helix of DNA relax. And then either strand is busy adding the correct nucleotides. Nucleotides that come from where? Yes, thank you. Food. From our diet. And so we're going to need a lot of them, as you can imagine. This is a huge, huge molecule. So each strand can act like a template. So in the end, we're trying to copy the DNA. Each strand can be involved in helping, now we got a new enzyme that's mentioned here, helping add the correct nucleotide. So here we see DNA polymerase. It's a good name for it. Polymer means a long repeating molecule. ACE indicates it's an enzyme. And DNA, well, what are we making after all? DNA. So that might help you remember. DNA polymerase. Helicase helps unwind. DNA polymerase helps to add the nucleotides. And they're being added one by one as the DNA polymerase works its way down. It's adding the correct nucleotide in place. So you have to have the nucleotides there to be able to do it. So it wouldn't happen without that enzyme. So we must have that enzyme in every one of our cells to be able to carry this out. Any questions so far? Just make sure you all are with me. So it's actually a bit more complicated. But there's these wonderful animations. So let's watch the animation and see a representation. And it's also very fast, too. And that's what I like about this animation. It kind of gives you a sense of, the zippiness of this whole process. DNA replication.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So in the animation, we were seeing that the double helix was being fed in, and then the nucleotides are being added. But here's the deal. DNA polymerase is an excellent enzyme. I'm very impressed with it, but it has a limitation, is that it can only add to the 3' end. Now this is where I needed you to know about 3'. Do you remember 3'? What did that refer to again? It referred to a carbon in what part of the nucleotide? Phosphate? Nitrogenous base? Or was it sugar? Should we go back and look? Maybe so. So let's go back and take a look at that 3' I was talking about. It was the sugar. So that they use this notation to indicate ends because we have it's anti-parallel. And so here is the 3' end. And so DNA polymerase would have no problem adding a nucleotide to the 3' end, but it can't do the 5' end. So that makes it a bit tricky. So how does it work then? So what it will do is that the side that it can do fine, it can add to, this is the 5' end, it can add to the 3' end. So it just can keep adding. This would open up more, and it would just keep adding. So it can add bases just fine, nucleotides just fine, this direction. But this direction has to be done in pieces. There is an enzyme, an RNA polymerase, that doesn't have that restriction, and it can add pieces of RNA, 3' or 5' doesn't matter. So it kind of gets things started. And so it seems like a very painfully complicated process to do this, but it's in all life, so it works. No other way has been found in any other living thing. And so what happens is they make, all cells, make little RNA primers, and then they can add to it. And so when this opens up a little bit more, we'll make another RNA primer and add to it. Anyway, what I just want you to know, bottom line here, is that this side has to be done in pieces. This side can be done continuously, no problem, but this side needs to be done in pieces. And eventually, you take out the RNA, and then you seal these with another enzyme. It's called ligase. So what's happening here is that we are adding nucleotides to each of these strands. Where there was one, there will be two. And they will have the genetic information because one strand can serve as the template for the other strand to be made. So it's made discontinuously in pieces that are sealed together. I want to pause just to see if you have any questions, because this is the part of the lecture I was not looking forward to giving, because it's so complicated. So if you have questions, we can also look at the animation one more time. Would you like to do that? Okay, I got a yes on the animation. So let's watch the animation. Now that you know what happens in pieces, you can watch for that.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So the end result is two new DNA molecules. Now, DNA polymerase is a fine enzyme, despite its inability to go 5' to 3'. It's a fine enzyme because not only does it copy, it does proofreading at the same time. Kind of like our computers. You're typing and it's telling you, don't you mean to put E before I in there? Oh, yeah. So it's checking as it's going along. So coming back to your question, Mateen, take a look at this little gem over here. There was a G, and what was added? T, problem. So the DNA polymerase has a proofreading function, and it checks. And not only does it place, and it's doing it so rapidly, too. That's why I like that animation. It's also checking it. And when there's an error, it replaces it with the correct nucleotide. That's supposed to be a C. So it's been repaired. Sometimes there is a buckling that happens when you have two T's next to each other. And so they're somewhat attracted to each other. So sometimes there can be mistakes. And so those are corrected as well. So it's not only an enzyme, but it's a proofreading enzyme. And so when they were coming up with how DNA copied itself, they think of 3 different possible ways that it could be. It could be that we have the DNA double helix, and then what results is that the new strands would have one of the old strands and one new strand, and that would be called semi-conservative. Or it could be that when DNA copied itself, that there was a completely new double strand that was made. So when you have 1 going to 2, 1 was the original, and 1 is completely new. And then there's the combination, I guess. It could have been a dispersive model where pieces of the new one are attached to pieces of the old one. But as it turns out, it's which one, of course? Semi-conservative. So we have a DNA double helix. The sides come apart. Bases are added to either of them, and they create the 2 new molecules that result. So anyway, if you see that semi-conservative, that's what it's talking about. It is the actual way that DNA copies itself, which is the point. For DNA, where there was one double strand, that there is two double strands. Where there is one chromatid, it will become two sisters if you continue long enough in that process, that amazingly long molecule. And for the most part, it's pretty darn good. But I've got the asterisk there because, despite the accuracy of the enzyme and despite the fact that it proofreads its work, sometimes there are mistakes that happen. And so those would be called mutations. And it's bound to happen because we're talking about volumes. Like floor to ceiling in this room, let's say a telephone books. That's the amount of information we're talking about. So let's say I gave you a job. I couldn't pay you enough, I don't think. But let's say I paid you $100,000 to copy telephone books from floor to ceiling as long as you did it accurately. I bet you couldn't do it. I know I couldn't. It's just every single letter, every single punctuation on every name and every phone number. It's an enormous amount of information. It's amazing that DNA doesn't make more mistakes than it does. And so it's just, because we're talking about just such an enormous amount of information, there is errors that are going to happen in replication. And because it's the code, that means that if the code is changed, like for example here, we've got, actually, can you see the mistake? I don't want to point it out. Where's the error? Guanine and, it shouldn't be pairing to adenine. So it should have had a cytosine put in there, but it had an adenine put in there. And so as we'll see shortly in the rest of this chapter, these nucleotides code for amino acids in protein. We're making protein. Then this code codes for proline. This code codes for threonine. And that could be a pretty big problem. Sickle cell anemia is based on a similar change. A whole different blood shape because it was an important one. So it could be extreme. Let me use an analogy of a book. And let's say you're reading this book and you're getting to the conclusion, and instead of trying to think of a good analogy, instead of the character dying at the end, it says not. Wouldn't that change the whole book? So if it's an important spot, the word not could really change the whole meaning. So it depends on where the mistake is happening. It could be just like, a minor mutation where, there's some just unusual striping pattern that happens instead of the spots in the back of this cheetah, I think it is. Or it could be more major. They have seen mutations like this. Do you know what's wrong with this fruit fly? Yeah, it's got too many wings. Just one little mutation can actually, depending on where it is, it can cause a dramatic change, and it could be life or death, like in this frog here. So mutations happen. And so mutations can be happening in what are called the body cells or somatic cells. And when they do, they can have a dramatic effect on the person's life. It's kind of hard to look at, but I just wanted to bring the point across. Skin cancer is pretty common, actually. So UV radiation can, or just simple errors, can cause the sequence of DNA nucleotides to change from what they should be. And then that can cause, as you know, depending on what gene is affected, it could cause cancer, uncontrolled cell growth. And this would be of great impact to the individual who has this cancer, no doubt. But I just want to point out that this doesn't have an evolutionary significance. Because if you get skin cancer, can you pass that to your children? What do you pass to your children if you were to have children? Ladies? Eggs. Men? Sperm. Right, you're just passing gametes. And so where's the mutation is really, that's a significant question. So if a mutation happens in what's called the germline, it's kind of an unusual name for it, but what germline refers to is the line of cells that undergo meiosis. I didn't call it that before, but that's the name for it, germline cells. Whether it be making sperm or eggs, it's the cell that you start with. So if the mutation's there and it undergoes meiosis, then the sperm are going to have the mutation, or the egg are going to have the mutation, like you see on the left side of this figure here. Germline mutation in sperm would then mean that there be a mutation that would result, every one of their cells would have that mutation, because that's the original, that's the DNA that you start with, the zygote. So every one of their cells would have it, and they would even pass it on to half of their gametes. But if a mutation is an area of the body that doesn't have anything to do with gametes, then it's not passed on. So lung cancer, for example, you wouldn't, if the exposure and the error happened in this individual, then that wouldn't be passed along. There is hereditary cancers, but in this case, we see that the mutation is happening here on this individual. So that's why when you get X-rays and things, they're always covering, your important gonad area, because they don't want you to expose it, to any offspring. But, mutation's important too, and we'll get that into another chapter, is that without mutation, there wouldn't ever be anything new. So a mutation is truly a mistake. But without these mistakes,